Hey, good evening, everyone. I hope you all are doing well and have had a good week so far. Today we'll be covering chapter 14. It's about Mendel and basically his gene experiments that led scientists to discover even um, more things about the genes and how inheritance or heredity works. So we're gonna basically start looking at what Mendel did and how his experiments kind of gave us clues, but how genes actually are passed on, and how basically traits such as height, eye color, for example, are could be looked at how they're inherited basically. Okay, so basically, during Mendel's, his name is Gregor Mendel, okay, time, he depended on the garden pea plants. So these are basically the plants that give you those green peas that you buy from the store. So he grew basically a lot of these plants, and he depended on them to be able to conduct many experiments that gave him clues about the gene interactions. So he looked at basically the flower colors of those plants. He looked at um, the peas of these plants, which are basically the seeds of them, their color, their shape of the seeds, all of these things that we're gonna talk about are today. Are we're gonna talk about the relationship of these traits and the genes. Okay, so let's just take a look at this. That's like a summary of the beginning of his experiments, and then we'll dive into like what happened in each step and why that happens. Okay, so this stuff is related directly to really the meiosis lesson. So meiosis, as we know, is what is the formation of gametes. Okay, so it's how genes are passed on from parents to offsprings. Okay, so this is what Mendel specifically wanted to see, like how things are passed on from parents to offsprings. Okay, so if we take a look at this one right here, you'll notice that there is a purple pea flower here on the left side, and then there is a white one. Okay, and you'll notice that so there's like many steps going on here, right? So you have basically the first one, which is this would be like his first offspring from these two flowers, okay? And then that offspring also is able to obviously produce gametes. So each one of these parents are producing gametes, right? Sperm and eggs, like we already learned in meiosis, okay? So we're learning how the sperms and eggs, they segregate or they separate. Okay, so when a diploid cell produces four haploid gametes at the end of meiosis, so we're learning how that combination contributes to variations, right, in offsprings. So in this first offspring, right, we will notice that Mendel didn't get like um, purple and white color flowered, right? So he only got purple flowered, and basically the offsprings of that came out of this original offspring basically had even different results. So let's take a look at each one and you know what it has, basically the content of each step, and we'll learn this together. Okay, so let's see here. Uh, so yeah, so we said that Mendel used what? Peas. So he planted them in his garden to plan his experiments accordingly. Okay, so this is a picture of Mendel between other scientists. Okay, so Mendel called the heritable feature, such as, for example, flower color. Okay, uh, that varies among the different flowers, he called them character. Okay, so another example would be, for example, talking about, for example, us humans, right? We could say that the height is a character. Okay, so each variant for a character, so if there's variation in the character, so in the case, if we're talking about flower color, okay, so each different characters, okay, we'll call this trait, okay. So you could have a trait that is, you know, purple or white, right? Another trait could be, for example, you know, talking about, um, you know, the peas, right? So if they're wrinkled or if they're round shaped, okay? So what are some advantages of Mendel using the peas, right? So why didn't he use, for example, other things? Why not human, for example? So peas actually have short generation time, so he could basically grow seeds and have plants very shortly, right? And he could get a lot of plants and a lot of peas to be able to work with them. And you know, in science, though the bigger the sample it is, the better the experiment, and the results that you get are more like, you know, verifiable and dependent on. Okay, so that we can depend on. So um, also, the peas specifically, the pea plants, Mendel was able to control them by the way they self pollinate. Okay, so he could basically do crossing of two different plants if he wanted to, or he could also let one plant, pea plant, self pollinate, and we'll see why that is in pea plants. Okay, so in pea plants, right, a plant has the ability to both have sperms and eggs. Okay, so it's able to self-pollinate and produce seeds, and basically, you know, keep producing offsprings that way. Okay, so what Mendel, you know, wanted to do first is to manually, okay, cross-pollinate two different plants, and because that's the reason why he wanted to, you know, do this experiment. He wanted to see how the color of flowers is changing when, you know, offsprings of these plants are being made. Okay, so in order to do that, what he did is he cut the the stamens, okay, from the purple flower, okay, and what he did is he um, allowed the stamens from another plant which had basically white flowers to pollinate that that purple flower where he cut those stamens. So stamens are the ones that give off the sperms, and the carpel is basically where the egg is located. Okay, so basically, if this was not cut, then the sperms from the stamens will basically pollinate the egg in the carpel, and you know, this, for example, this purple can self-pollinate. But so Mendel wanted to basically manually cross um, you know, different colored flowers to be able to see what kind of offsprings does he get. So after crossing it manually, okay, let me just tell you that so since these are the parents, if you will, so this would be the parental generation. Okay, so this is denoted as P here. So he wanted to see what kind of offsprings, okay, that he would get. So the offsprings he would get when these peas, right, that are products of the crossing here, they grow and become plants. Okay, so the results of these peas showed that the first generation offspring, or filial generation, we call these F1 generation. Okay, so these are the first offsprings from these parents. Okay, so what he noticed is that all of the plants were or had purple flowers, okay? Although one of the parent had what? White flowers. So he was like, what is going on here? Did the white color just disappear, or what's going on here? So this was the beginning of his experimentation. Okay, so let's go together in this and see what happened next. Okay, so um, first of all, Mendel picked what we call true breeding here. So the plants that he picked, okay, were basically followed for many generations, and basically he made sure that these um, either purple flower or white flower are called true breeding, which means they've produced basically the same flower color offspring for many generations when they self-pollinate. So as these automatically self-pollinate if Mendel didn't cross them. Okay, so he just followed and saw that this specific flower, right, was able to self-pollinate many generations and produced purple color, and same thing for the white one. So these would we call them true breeders. Okay, so the true breeders in this case are what? The parents. Okay, so they're the parental generation, and so the hybrid offspring that are from the parents are the F1 in this case, F1 generation or filial generation 1. Okay, so um, when he mated those parents, right, the process what we call is hybridization. Okay, so that is the crossing that he did between the two true breeding varieties, the purple and the white flowers. Okay, now when the F1 generation self-pollinate, okay, so he didn't touch those anymore. Okay, so he let them do what they need to do automatically. Okay, so F2 generation was produced. Okay, and we will see what the F2 looked like. Okay, so this is just talking about the same thing. So one thing is that before we talk about the F2, so let's just talk about one thing in the F1. So when he crossed the purple and the white color flower, and he got purple flower only, okay, back then during the 1800s they had this hypotheses, okay, so hypotheses, which is what, it's basically a question that, or something that they had in mind that basically they were expecting to have as a result of the crossing between the white and the purple flower. So they were expecting to have the blending hypothesis, which means both the white and the purple would produce a blended color, because they're being blended together basically. So but that's not what happened. You notice that like for example red or pink or whatever was not produced, which is like the blending, if you will. Purple was only produced, which is the trait that was seen in the one of the parents. Okay, so so now let's take a look at the F2 plants. So after these basically self-pollinated each one, okay, so they allowed themselves to produce seeds, and the flowers or the plants were grown from those seeds. Okay, Mendel saw that the F2 plants had actually a mixture of purple flowers and some were actually white again. After he noticed that the F1 were completely purple, the F2 actually had a combination now. Okay, so there was a ratio though. So he noticed that for every one, for the whole results that he got, basically there was a ratio of 3 to 1. So for every 3 purple flowers there was 1 white flower. So he got basically 1,000, the total of 1,000 plants, I believe it was. And so about 700 of those were basically purple plants, and then 300 were white plants. So that would be like 3 to 1 ratio. Okay. So this is just showing you basically the same thing again. So these were the parents, okay? Then from the parents, after Mendel crossed them manually, right? Purple flowers were obtained, okay, as you remember, right? So now he allowed these to self-pollinate, okay? So after these were self-pollinated, the F2 generation were what? 3-to-1 ratio, as I told you, produced. Okay, so you could see that the white color disappeared in the F1 generation, but it reappeared in the F2 generation. So Mendel was like, what is going on here? Why did this happen? Okay, so you can watch these videos that kind of explain, um, you know, a lot of things in an animated way. Okay, so basically Mendel reasoned that the purple flower, okay, was basically um, the only factor that was affecting the the color in the F1 hybrids. Okay, so that is why the F1 offsprings were completely what? Purple. Okay, so because of that, Mendel called the purple flower what? A dominant. Okay, so it dominated the white color. So we call the white color in this case because the purple is dominant. The white one would be the recessive trait. Okay. So as I told you, so the white color was not destroyed in the F1 generation, although only purple flowers showed up in Mendel's experimentation. So the we know that it was not destroyed or diluted because what? They showed up again in the F2 generation. Okay, although it was a ratio of 3-to-1, but they still showed up. Okay. Okay, so here it's just saying that Mendel also observed not only he studied the flower colors, but he also studied the peas of the plants. So he studied the color of the peas, he studied the shapes of them, if they were wrinkled, if they were like rounded, smooth. Okay, so what Mendel called a heritable factor. So he called it a heritable factor. That means the material that was being passed on, okay, from the parents to the F1 or the F2 generation. He called it heritable factor back then. We now know it as what? A gene. Okay, what do we know about a gene? We know that a gene is a part of a section of a or a section of the DNA, right? So a gene is made out of many sequences, right, that identify that specific gene on the DNA. Okay. So these are the things that basically Mendel was able to, all the different characters or traits characters that he was able to study about the pea plants. Okay. So Mendel developed a hypothesis that explained the 3-to-1 ratio that he got in the F2 offspring. Okay. So there was basically four concepts that basically he formed based on what he observed from the F2 offspring and how basically the white color disappeared in the F1. Okay, so let's look at his four concepts. Okay, um, that he was studying. Okay, and looked at. So the first one would be that alternative versions of the genes, okay, accounts for variations in inherited characters. Okay, so for example, the gene for flower color, right, in pea plants exists in two versions. Okay, one for purple flowers and the other for white flowers. Okay, what are we talking about here exactly? So we're talking about the what? Homologous chromosomes, right? So one would be a purple, right? And the second one would be what? White flowers, right? So that is in the parents before basically they go through meiosis, which basically separates the gametes. Okay, so these alternative versions of gene are called alleles. Okay, so alleles are, you know, basically located at specific. We call it locus. Okay, locus is basically a specific location. So it comes from the word location. Locus comes from the word location. Okay, on a gene. So it's a specific location on a gene where you will find an allele. Okay, so an allele will be basically a variation in genetic coding, if you will. Okay, that codes, for example, the allele would be um, for example, flower color in this case. Okay, so you could, for example, have a dominant allele or a recessive allele. Okay. So now let's take a look at what I just kind of explained briefly. Okay, so this is what we're talking about when we're talking about the first concept of Mendel. So it would be the talking about the alleles, which are alternative versions of a gene. Okay, so if you remember, right, the homologous chromosomes and the um, diploid cell that's producing gametes. Okay, so one would be from what? Would be from each parent. Okay, one from dad, one from mom. Okay, you will notice that at a specific location or a locus, okay, there's gonna be a gene. Okay, and in that gene, it's an allele, right, that codes for what? For example, purple flower, right, in the case of this. And for example, the second chromosome that's next to it, right, across, you can notice that the location would be exactly across from it. Okay, the allele for the one, let's say from the mother, would be what? It's allele for white flowers. Okay, and that's how it looks like if we're looking at the actual DNA sequence. Okay, because remember, chromosomes have what? Condensed DNA in them. Okay, so you can notice that the alleles for purple flowers, okay, has a different base than the allele for white flower. So this allele right here allows the formation of an enzyme in this example. Okay, that enzyme basically helps synthesize the purple pigment in the case of, if we're talking about purple flowers. However, because this sequence is different, okay, it does not code for this enzyme that forms the purple pigment. So as a result, we get what? No purple color or white color instead. Or instead, you could get basically a recessive allele, okay, and so it wouldn't show up because if they're both together, because why? Because this would be the dominant. Okay, so the second concept that Mendel basically created was for each character. Okay, so we're talking about, for example, the flower color. Okay, an organism inherits two alleles, one from each parent. And this is what we just said also here, kind of. Okay, so not only he said that there are basically alternative versions of the genes. Okay, because of different chromosomes. Here, he's saying that each actual offspring inherits two alleles, actually, not just one from each parent. Okay, so he didn't know about chromosomes back then, obviously, back in his time in the 1800s. Okay, so um, I'm just reading this to see what's kind of important to tell you. So these alleles could be similar, right, or they could be different, basically, right? So if they're similar, we call them what? We call them homozygous. Okay, so we're gonna talk about this in a bit, but if they're similar, we call them homozygous. If they're different, we're gonna call them heterozygous. Okay, so in the case of purple flower, right, we said that the dominant allele is gonna basically win in color wise. Okay, so but Mendel didn't know if the purple color was homozygous on both from both homologous chromosomes or heterozygous, which is basically they're different. One is a dominant and one is a recessive gene. Okay. So the third concept, okay, is that if the two alleles at a locus differ, okay, so this would be what? So this would be the heterozygous. So one would be the dominant, okay. So one would be the dominant, determines the organism's appearance, right? In the case of purple flower, that's what happened, okay. And the other would be basically the recessive, as I mentioned, okay. So let's say this would be the dominant, this would be the recessive, okay. Now the fourth one, and his fourth concept, he talked about the law of segregation, okay, and this says that the two alleles for a heritable character, okay, we're still talking about, let's say, the color of the flower, okay, he's saying that these two alleles separate or segregate during gamete formation, and this is what we know, right? We know it as assortment, independent assortment, right? We studied this in meiosis. So this means that the genes basically from the diploid cell separate, when they're forming the gametes, the haploid cells, okay? I'm just giving you a relationship between Mendel's experimentation and what we studied in meiosis, okay? So, thus, an egg or sperm gets only one of the two alleles that are present in the organism. Is that what happened, right? That's what happened. So, we started with a diploid, right? We get a haploid at the end, which has what? Instead of 46 chromosomes, we're getting 23 chromosomes, okay? So this segregation of alleles corresponds to the distribution of homologous chromosomes to different gametes in meiosis. This is very clear, right? Okay, so let's take a look at this again here. So now we're basically, we know kind of what's going on. Okay, so the model accounts for three that Mendel was able to see was that in the F2 generation he was getting this 3-to-1 ratio that we talked about. So three were purple colored and one was white flower. Okay, so we use this square right here, which we call Punnett square. Okay, and this square allows us, right, to be able to see a possible combination, a probability of the combination of genes, and these would be the offspring's probabilities. Okay. So what you will notice is that in the P generation, this true breeder was a big P, and that is saying that it's got basically two dominant genes on those homologous chromosomes. Okay, this one has small p, which is also a true breeder. Okay, so do you notice that the true breeders cannot be big P together? Okay, they have to be what we call homozygous. So these are homozygous because they're the same. These, the big P, we call heterozygous. So hopefully you're understanding this by now. Okay, so basically the gametes of each flower, the purple and the white, so you're getting basically one gamete out of the two, right? And one here. So when the big P, small p mixes, or basically gets fertilized, okay? And so in the case of flower, they're pollinated, okay? So we're getting the purple flowers as a result in F1 generation. This is what Mendel got in the F1 generation. Okay, so it would be a big P, small p. So that means the gametes of this one that's able to be produced is half of them would be big P, half of them would be small p. Okay, so let's put the gametes on the Punnett square. So the gametes will go on the outside of a Punnett square that's basically produced from each parent. So since basically these purple flowers are self-pollinators, so we're going to use basically big P, small p twice as basically the male and female in a single plant. So we'll put them twice, one on the x-axis and one on the y-axis, okay? And we'll basically combine them in each in each square here, okay? So it would be big P, big P, big P, small p, big P, small p, double small p's here, okay? And since the purple flower, which is a big P, is the dominant, anytime you see a big P or big P, small p, as you can see here, they will be always purple flowers because the purple color is the dominant. If you see basically a small p, that's saying that, this is recessive, right? And that is why it's white. And so that is 3-to-1 ratio, okay? And this is what we're talking about when we say homozygous, right? It's when the two genes are exactly the same. They're either both of them dominants or both of them are recessives, as you can see here. If we say heterozygous or heterozygote, right, we're saying that the big P is with the small p, so they're both different. So one is a dominant, one is a recessive together. But the color that you see, or the phenotype, this is what we call it. We call it what you see with your own eyes is the phenotype, or the expression of the genes, okay? So this would be basically purple because the big P is the dominant, okay? Okay, so this is just saying that organism's traits do not always reveal its genetic composition. Why is that? Because, a purple flower could either be a homozygous, which is big P, or a heterozygous, big P, small p. So that means its genetic or genotype information, genetic makeup, is basically, different, right? So we won't, if we see a purple flower, we can't just assume immediately that it's a homozygous or heterozygous, okay? So the phenotype, we said, is the physical appearance or the expression of the genes or the genotype of an organism, okay? So this is just showing you the phenotype, okay? So the phenotype is what you see, okay? We were able to see in the F2 generation that there was 3 purple colors, one white flower, and then in the genotype, however, it's different because as I mentioned, the big P or homozygous is purple, while also the big P small p is also purple. So that is why there's difference, okay? Okay, we talked about this. Okay, so in order for us to determine the genotype, let's say we're talking about this purple flower. We know that the purple color is the dominant, okay? But we don't know if this flower has a genotype of big P or big P small p. So whether it's a homozygous or a heterozygous. Okay, so we will do a test cross, okay, to breed like offsprings, okay, and see what do we get to be able to find out if this individual flower, has a big P or a big P small p. Okay, so if the offsprings, right, display the recessive phenotype, which would be what, the white color at all, then we will know that this purple is actually what is actually the heterozygous. If it doesn't, if all you get is purple, then basically, that means that this flower has big P, or homozygous. Okay, so let's see. There's two types of predictions here. So if we predicted that this flower has big P here, as you can see on the left side, we put them right here. So the eggs would be big P, and we crossed it with a small p. Okay, so the result would give you big P, four of them, and all four would be purple colors, because the big P is, dominant for purple color. Okay, now on the right side, if we say that the genotype is big P small p, okay, and we put them right here and we try to crisscross them, okay, we will get white flowers. Okay, so that means this is a genotype. Is that what happened with Mendel? Not exactly that, not 50-50, but he did get 3-to-1 ratio in the F2 offsprings, which tells us that the initial parental purple flowers were big P small p. Okay? No, not the initial, sorry, the F1 offspring. Okay, so the original ones, remember, they were a true breeder, so big P. Okay, sorry, I was thinking about something else, but so the F1 generation, okay, after they basically were self pollinators, okay, they produced basically the white colors, which tells us that the F1 generation is big P small p, after they were, after the parents were basically crisscrossed. Okay. Okay, so Mendel looked at a single trait, and this is what we call monohybrid. So it's basically following with just one character across. Okay. So a cross between such heterozygotes is called a monohybrid cross. Okay. Okay, so now we're gonna also look at the dihybrid cross, which basically it's showing us two differing characters. Okay, so we could be talking about, for example, flower color, and for example, the shape of peas, for example. Okay, so let's see what kind of dihybrids we're gonna look at right now. Okay. So a dihybrid cross, right, is a cross between the F1 dihybrids, so not the parental, but the F1 plants. Basically, they can determine whether two characters, basically, as I said, either like, we could talk about just the P's in this case. So, P color and P shape. So these are two characters. How they're transmitted to offspring as a package or independently. So we will see what happened here. So let's take a look at this experiment that's basically looking at two characters now, or a dihybrid. Okay, so in the P generation, okay, so first of all, Mendel was asking, do the alleles for one character, okay, let's say, the color, assort into gametes. So when gamete is being formed, okay, is it dependently or independently of the alleles for a different character, which, let's say, we're talking about the shape of the P? So do they assort different, like the same together, or differently? So let's see. So if we take a look at this P, okay, and which is basically what, a yellow and round, and this P is a green and what, and wrinkled. So after the gametes are produced, so the gametes are produced would be what? One from each character. So because these are what? Homologous chromosomes, so you're gonna use one of them. So it would be big Y and big R in this case, which are what? The dominants in this case. So why are they dominants? Because they're capital letters here. We're using them to indicate that they're dominants. Okay, so we know that the yellow color is dominant. We know that um, the big R or the round P is a dominant trait. Okay, so here in this other one, right, the gamete is small y and small r, indicating that it's not yellow but it's green instead. Okay, so it's recessive and it's wrinkled. It's not smooth round, so it would be also a recessive trait. Okay, so small y, small r. And then when these gametes are crisscrossed, right, together, you get the F1 generation, which is basically a combination of the two. So big Y, big R, small r. So this would be a what? Heterozygotes. Okay, so let's see here what happened next. Okay, so we're still asking, you know, if the alleles for one character assort into gametes dependently or independently of the alleles for a different character. Okay, so we're looking at two different things here in this in these tables, on these Punnett squares. So the left one would be the hypotheses. So it's only a hypothesis of dependent assortment. Okay, which is saying basically that they're both dependent and they will assort together. The right one is saying the hypothesis of independent assortment. Okay, we'll see which one is actually the correct way to go about this. Okay, so let's take a look here. So we're looking at the predicted offspring of the F2 generation. So this was the F1. So the F1 is going to produce gametes, and we're going to put the gametes on the outside of the Punnett squares. Okay? So in the case of the male, okay, what we're going to get is what? Big Y. Let me just show you. Keep going back and forth just so you can see. Big Y, big R here, and small y, small r here. So half, half, right? Same thing for the eggs. Because why? Because it's, you know, self-pollinating here in this case. So the result shows that, you know, you get big Y, big Y, big R, right? And you can tell what the phenotype is because of, you know, we know which is the dominant allele in this case. So the result shows that it's a 3-to-1 phenotypic ratio, kind of like what Mendel got with the flowers, with the flower colors. Okay, didn't he get 3-to-1 in the F2? Same thing. So let's look at the hypotheses of the independent assortment now. Okay, so let's say that the gametes that were produced, okay, there's an independent assortment. So that means they're assorted more differently. So the big Y will combine with the big R to produce this one. The big Y will combine with the small r to produce another gamete. The small y will combine with the big R to produce another gamete. And finally small y with the small r to produce this gamete. And the same thing happens again on the egg side, because why? It's self-pollinating. Okay, so this would be the results that Mendel, you know, would get. So you will notice that 916, so there's 16 results here. You could count them, right? So 4 times 4 is 16. Okay, so what he saw was there was a 9-to-3 to 3-to-1 phenotypic ratio that he noticed. Okay, and so this is what actually happens, and this is the reality of dihybrid cross. Okay, so this is how the alleles, okay, they assort independently based on the results that Mendel was able to get. Okay, not this one. Okay. Okay, so based on the dihybrid cross that and the results that Mendel was able to get, he developed the law of independent assortment, and we are still using that where in our lessons, right? We learned about it in meiosis. Okay, so it states that each pair of alleles segregate independently, okay, of any other pair of alleles during gamete formation. Okay, so the allele that codes, for example, for the shape of the seed is gonna basically assort independently that for of another allele that codes for the the other character, or the shape or the color of the seed. Okay. Okay, so again, this is what basically uh, Mendel was able to come up with. So the law of independent assortment, is showing that the different characters or different alleles are, independently assorted. Okay. So we're going to talk about a little bit about the rules of probability. In order to be able to like calculate probabilities of genes during gamete formation and offspring formation. Okay, so basically, when tossing a coin, right, the outcome of one toss, right, when you toss a coin, has basically no impact on the next toss, right? So that is the same way the alleles of one gene, okay, segregate into gametes independently of another gene's allele, as we said, right? So that's the law of independent assortment. Okay. So there's basically, now it's talking about the um, probability rules, okay, again. So it's going to use a little bit of what? Math, okay? I know many of us don't like math, but we have to study about this here. So the multiplication rule basically states that the probability, so what are we doing? We're multiplying, that two or more independent events will occur together is the product of their individual probabilities. So there is a multiplication, okay, that would give you the product, and that is the multiplication rule, okay? So the probability in an F1 monohybrid cross can be determined using the multiplication rule. Let's see how that is, okay? So the segregation in a heterozygous plant, right, it's like flipping a coin. So in the case of the purple plant in the F1 generation, right, it had what? It's a heterozygous, so it had a big P and a small p, okay? So there's a half chance of carrying the dominant allele, which is the big P, and there's a half a chance of carrying the recessive allele, okay? Do you see that? So if we have here, big R, big, small r, but it's the same thing, right? Big P or small p, same thing, okay? So it's half chance, okay? So the way we do this is what? If we multiply these two, we will get the probability, which is what you see here. So half times half is 1/4. Okay, and the same thing here and here and here, right? So that is the multiplication rule, okay, of this. So now the second probability type of rule is the addition rule. So there's now addition instead of multiplication. Okay, so states that the probability that any one of two or more mutually exclusive events, right, will occur as calculated by adding together their individual probabilities. So we're adding right now, okay? That is why it's addition rule. So let's see what that looks like. So the rule of addition can be used to figure out the probability that F2 plant, okay, from a monohybrid cross, so we're talking about monohybrid cross here, will be a heterozygous rather than homozygous. So this is kind of exactly talking about if you look at the the flower colors, we're trying to see what is the probability that the F2 plants that Mendel was getting. Remember, they were what, 3-to-1 ratio. What is how did that probability come about? Using the addition rule. Okay, so let's take a look here. So that would be basically what, adding, right, together. So the addition rule will say that you're adding to be able to get um, the not the product, but the result you're looking for. Okay, so I don't have here to show you an example. Actually, this is the same example. Yeah, I probably added the wrong one here. But anyways, the addition is adding basically to figure out what you're gonna get as a result. Okay, so we can use basically the rules of probabilities like multiplication and addition to predict the outcome of crosses. I think I have something here to show you probably. Okay. Let's see here. Okay, so let's take a look at the probability of big Y, and big R. So these are what homozygous dominance for both, the yellow color and then the round shape. Okay, so let's just see here what that looks like. Okay, so the probability of big Y is 1/4, and the probability of big R is 1/4 as well. Okay, so let me see here what we are looking at. Okay, so let's see here. Okay, so this is talking about this one, which is, okay, so if we look at this big Y, and big R, I'm trying to remember what part of the lecture I need to tell you about here. So the big Y, and big R is talking about this specific square here. Okay, and it's telling you what is the probability that you would get this type of result from these crosses here. Okay, so 1/4, which is basically a big Y, and 1/4, okay, if you multiply them, okay, you will get what, yeah, there's a typo here actually, so it would be, actually no, that's correct. So big Y, big R, right? You will get 1/4 times 1/4, you will get this 1/16th, which is what you see here, right? So there's 16 squares here. So the probability that you would get this specific genotype and phenotype, right, is 1/16th. Okay? You can't say phenotype because phenotype actually has several here, but let's just say specifically genotype. Okay, sorry about that. So genotype is 1/16th. Now let's take a look at this second one that we have here as an example. So if we're talking about big Y, small y, so let's find where that is. Big Y, small y, and big R. Do you see it? Okay. And do we have more of these? Yes, we do. Look, big Y and small y, and big R. Okay, and that's it. So let's see if that's actually correct. Okay, so if we look here, we have basically big Y, small y, okay, which is 1/2, right? And then basically you have 1/4, which is big R, okay? If you multiply them, which is, this is a multiplication rule, right? You'll get 1 on the numerator, and 2 times 4 is 8. So that is the denominator, okay? So that is what we get here, basically, okay? So we get 1/8 probability of this specific genotype showing up in this Punnett square. So these probabilities are not easy, as you can see. So they require a little bit of math, and I was trying to figure out if we're talking about um, the results, or the actual like different things. But this is just talking about each genotype that's found in this Punnett square. Okay, and it's we're calculating the probability of each one. Okay. Okay. So let's take a look at the probabilities, right? In this specific one, so that is a tri-hybrid cross. Okay. So there is how many characters? 3 characters now instead of 2. So it's getting a little bit tougher here. Okay. So what are the probabilities if you have at least? So it's asking for the least 2 recessive traits in the following cross. So let's take a look here. So we got a big P, small p, big Y, big R, small r. So that would be what a heterozygous for all 3 characters. Okay, now this one is actually contain different things. So this would be a heterozygous for the big P, and small y, and small r for these homozygous, and this is a heterozygous for those alleles. So let's take a look here. So big or small p, small y. So if we're mixing basically this with this small p with this small y, okay, so you have to basically segregate. This is not showing you here. I should have shown you a little bit more more detailed workup how to do this. Okay, so but this is basically how to do the probability is to basically use the multiplication rule. This is just showing you how the multiplication rule is being used to get the probability that you would get in a Punnett square of this specific trihybrid cross. Okay. Okay, so again, the relationship between genotype and phenotype is not as simple as what happened in Mendel's studying and experimentation. It has more complexity to it. Okay. So many heritable characters are not just determined by only one gene with 2 alleles, but it's more. There's more to it. Okay. So this is just saying that that the inheritance of characters by a single gene, okay, may deviate from simple Mendelian patterns, right? In the following situation. So let's see what situations does gene expression, or what you get out of the gene is actually different than what you would expect. So one of the reasons why you would have that is because when the alleles are not completely dominant, so they're not completely dominant, okay, or recessive, or when the gene has more than 2 alleles that codes for it, okay, or when a gene produces basically multiple phenotypes, okay, so that's when you would not be able to apply directly to Mendel's experimentation. Okay, so now we're going to talk about the dominance, or what we call complete dominance, okay? So that occurs when phenotype of the heterozygous and dominant homozygote are identical. So that's what happened in purple flowers, right? Didn't we get purple flowers whether it was a true breeder, which is, we know that it was what? A true breeder is always a homozygous for the dominant allele. So we knew it was big P, right? And we still got purple flowers in the F1 generation, which we knew that it was what? A heterozygote. So that is complete dominance of the, big P allele, okay? Now, in incomplete dominance, which is basically, this is what this point is talking about, when there's an incomplete dominance of that big P, the phenotype of the F1 hybrids, right, it will be somewhere between the phenotypes of two parental varieties. So, in the case of flowers, you would get actually a third color because, there's not complete, if we will, penetration of the color. Okay, so um, that would give you a third color, such as, like, for example, a pink color, okay, instead of either a purple or a white color. So let's see what's gonna look like in the next slide. But let's also talk about one more thing, which is called codominance. So codominance is different than complete dominance and incomplete dominance. Codominance is when two dominant alleles, right, are affecting the phenotype in separate distinguishable ways. So now there's two alleles, okay, instead of one, affecting the phenotype. So let's take a look at the incomplete dominance, which is this one. Let's see what that gives you when it's there. So let's take a look at the parental generation. So in this case, we're looking at basically a red color flower and a white color instead of a purple and white. Okay, so because we're talking about incomplete dominance, we're gonna denote the gametes as, a big C on both, and then we'll basically put the superscript R. That's just saying that the dominant allele is red, okay, and it's basically denoted as R, big R here, and this would be a W for white. So because of there's an incomplete dominance, okay, by either the parental generation, what we're getting is a pink color, so neither the red or the white. Okay, so you will see that, we will still write it as, two big C's and an R and a W on each one. Okay, so now the gametes of this pink flower is going to be what? Half of this and half of this. Now let's take a look at the F2 generation now. What happened is that in the F2 generation, if we put those gametes here, when this self-pollinate, so we're repeating the same thing for the eggs and the same thing for the sperms, okay, we're actually getting again some of the parental colors. Okay, this tells us that there's an incomplete dominance, okay, in the dominant alleles here. So they show up again somewhere in the later generations. Okay, so this is just talking about the wrinkled or the round P's of that character in the P's. Okay, so it's saying that in the recessive allele, right, which is coding for the wrinkled P's, as we saw, right, in the Mendel's experimentation. So when the recessive allele is there, it codes for a defective form of the enzyme, which leads to an accumulation of unbranched starch inside that pea. So because of that, basically the water causes water to enter the seed as a result, okay, which then wrinkles it as it dries. Okay, so you could see um, the effect of that gene on the actual pea itself. Okay, now this is talking about this Tay-Sachs gene, which is a fatal inherited disorder. So it is inherited, and it's a dysfunction in the enzyme that causes accumulation of lipids. We studied what lipids are, like, fats are lipids, right? Um, when this disease is manifested, you'll have basically a dysfunction in the enzyme that causes accumulation of lipids in the brain. Okay, so the allele for this disease is recessive. Okay, sometimes you could have a disease that is caused by a dominant allele, and we will see what examples we have in later slides. Okay, so at the biochemical level, the phenotype, that is the enzyme activity level, is incompletely dominant in this case. Okay, for this disease, at the molecular level now, the alleles are codominant. Okay. So this is talking about the frequency of alleles now in the population. So just because it's, it sounds like, like a good thing to be a dominant allele, it's not always necessarily the case. Okay, so sometimes there's more recessive alleles in the population than dominant alleles. Okay, so in the case of numbers of fingers and numbers of toes in humans, right? Um, well, when there's more than 5 fingers on one hand or toes, we call this polydactyly. Okay, so that is caused by a dominant allele. Do you see that? That is found much less frequently in the population than the recessive allele. So basically, if any of us have 5 fingers on each hand, okay, that would be basically due to the recessive allele. So that is basically saying that not all the time a dominant allele, is basically more frequent than a recessive allele. Okay. Okay, so most genes exist in populations in more than 2 allelic forms. Okay. So there's basically 2 forms of allele that's given us the phenotypic presentation. So as an example would be that the 4 phenotypes of the blood type, okay, which is ABO. This is what we call it. We call it ABO, blood group in humans, right? So that's how it's denoted. Usually blood types for a single human is denoted as basically 2 I's next to each other, okay? And if the person is basically AB, the I would have an A on it, okay? Basically that would be the IA allele, okay? And then the second I will be I and B, okay? So that's denoting that the person is AB positive, okay? So those AB are actual carbohydrates that are on red blood cells. Okay, and that's what that looks like here. Do you see that? So 2 I's are always presented as I was saying. Okay, as you can see here. So if the person has 2 A's on those I's, that means the person is A positive. Okay, so that person has, let's say, this triangular shaped carbohydrate on the surface of red blood cells. Okay, and if it's A and B, that person is AB phenotype. Okay, Now, if the carbohydrate is not present at all on the surface of red blood cells, which is basically a recessive, you can see it's a recessive genotype, then the phenotype would be O positive. Okay. Okay, so let's talk about now pleiotropy. Okay, so uh, pleiotropy is when most genes have have multiple phenotypic effects. Okay, so more than one phenotypic presentation. Okay, so for example, pleiotropic alleles are responsible for the multiple symptoms of certain hereditary diseases such as cystic fibrosis and sickle cell disease. Okay, so that means there are more than one allele, um, present in the disease, and so if one allele is basically working, okay, and it's not defective, the disease won't present as basically badly as if the both or more genes are basically like recessive, for example. Okay, so sometimes this could actually allow survival of an individual instead of death. Okay, so some traits may be determined by two or more genes. Okay, so in epistasis, one gene affects the phenotype of another gene due to interaction of their gene products. So that is a indirect effect of one gene onto the other. That is what we call epistasis. So they're different terms, okay? So please make sure that you learn the definition of each one and what it does. Okay, so in polygenic inheritance now, which is also a third term that needs, to be studied, multiple genes independently affect a single trait. So now we're going backward. So now we have multiple genes affecting a single trait. Okay? So that would be, I think, an example of height, if I remember correctly. So height is actually controlled by several genes that code for it. So not just one gene is going to code for how tall someone is going to be, okay? So let's talk about just epistasis a little bit, which is what? One gene affecting the phenotype of another due to interaction of their gene products. So the expression of a gene at one locus, as I said, alters the phenotypic expression of a gene at a second locus. So it's an indirect effect on that second gene. Okay, so for example, in Labrador retrievers, so that's what I think they're called. These are types of dogs. In many other mammals, coat color depends on two genes. Okay, so one gene determines the pigment color. Okay, so let's say big B is for big B is for black and small b is for brown. The other gene determines what if there is color or no color at all. Okay, so you could see how both of them are playing together in order to get the phenotypic expression. Okay. Alright, so this is kind of similar to what we were seeing in the Mendel's stuff. Okay, so I'm not going to go through it again. So polygenic inheritance is this, what we just said here. Okay, so there's multiple genes independently affecting a single trait. Okay, so let's see here. Quantitative characters are those that vary in the population along a continuum. Okay, so Quantitative, did I say qualitative? Maybe I said quantitative. Quantitative variation usually indicates polygenic inheritance. Okay, why? Because it's an additive um, effect, right? As I said, so basically many genes are contributing to the addition of effect of two or more genes on a single phenotype. So, if for example, five genes are contributing to height, the person will be basically taller. If we're talking about also like here is don't worry about this whole thing. You're not required to know this, but um, this is just showing you the different skin colors, okay, pigments. So you can see that there's many genes contributing to how light or how dark a skin color is going to get. Okay. So also I wanted to mention that there is another like completely different thing than the actual genetic makeup that determines how a phenotypic presentation is actually expressed. So that would be depending on the environment. So for example, the skin color, right? It could be dependent obviously on the genetic, as we said, right, in polygenic inheritance. Depending on several genes that basically use the addition of expression to be able to see, how dark its skin color will be. But also, you could expect that an individual that spends more time in the sun, right, is gonna get, a skin that's darker eventually, right, with time. So that is when we say there's an environmental effect on the phenotypic presentation as well. Okay. So the traits that depend on multiple genes, okay, which is like what? Polygenic inheritance, okay, along with or combined with environmental influences such as, for example, the sun in the case of skin color, that is what is defined as multifactorial. So there's multi-facts or causes that's causing the specific phenotypic presentation. Okay, so an organism's phenotype, obviously the way that that organism is presented or the way they look, okay, physical appearance, is due to the anatomy and physiology, right, of that individual, genetic makeup and behavior, and obviously the environment and the history that this environment and where that individual um, was basically in his lifetime or her lifetime, that determines the physical appearance. Okay, so that's just saying why humans are basically not a good subject for genetic research, because generation time is basically too long, and it's basically the opposite of the peas plant the pea plants. Okay, so again, Mendelian genetics, they could be used still to study human genetics. Okay, now in the case of humans, right, we have something called pedigree analysis. So this analysis will allow us to see the current um, genetic makeup and the possible future genetic makeups of like future basically generations. Okay, so pedigree again is basically a family tree that you could actually see and describe the inheritance between the different generations, from the parental generation to first, second, and third, and so on, right? Um, you could know and basically track down where a certain recessive or dominant gene you could track it where it started and all of that using the pedigree analysis. Okay, so and you can also predict future offsprings based on it as well. Okay, so what you're seeing here basically is we're looking at the widow's peak as an example here of this person. Okay, so there's actually two persons here. So so the widow's peak is this hairline right here. You see that peak? Okay, so this is just looking at the first generation, which are the grandparents. Okay, and then the second generation, okay, which are basically the offsprings of these grandparents, right? For both mom and dad, right? So which ones would be the mom and dad of these two sisters? These ones, right? So these grandparents are on the side of, for example, dad, and these would be on the side of mom. Okay. So the big W and small w are heterozygous, right? As we know. So the big W would be the dominant gene, and then the small one would be the recessive. Okay. So you could see that the whenever the widow's peak is basically phenotypically expressed, you will see that it's basically a purple shaded either circle or square. So square is for male, and then the circle is for a female. Okay. And so you will notice that the parents of these two sisters, right, were what both of them expressing the trait, okay, which is what the widow's peak. So this parent, which is the male parent, right, had a heterozygous, so big W, small w, and the female parent, right, the mom, had the big W, small w. So what they produced was half, right, half had either their offsprings had either the big W, right, or big W, small w, which is what also dominant. So this sister will present and express the widow's peak, while this one will not, because she received the recessive alleles. Okay, so you could see this is called the pedigree that allows us to analyze a certain trait. And this is also similar thing, but it's talking about like tasting, a chemical tasting. Okay, so this is basically talking about the same exact thing. So it's another example. Okay, so many genetic disorders are inherited in a recessive manner. Okay, so these would be like either mild or life-threatening, depending on how much the presentation is, excessive. Okay, so in the case of, for example, albinism, okay, that is actually recessively inherited, right? So the recessive inherited disorders, they only show up in homozygous for the allele. So the individual has to have both alleles as recessive. Okay. Now, when we say carriers, we're talking about what? An individual that basically carry the recessive allele, but does not show the phenotype or the trait of the dominant basically allele. So they could still pass it on, right? Such as, for example, when we looked at the purple flower, right? In the F1 generation plants, these plants basically still showed what? The purple color. However, the F2 generation showed white also. So that means the F1 generation was a carrier because they were also heterozygous. Okay? So most individuals with recessive disorders, okay, are born to carrier parents. Okay, why? Because the carrier parent has what? One of their alleles that is a recessive allele. That's why they're heterozygous. Okay. So albinism is a recessive condition, right, characterized by a lack of pigmentation in skin and hair. Okay. So let's just take a look here. So a normal phenotype parent with a normal phenotype parent, big A, small a. So these are both heterozygous. Do you see that? So that means they're what? Carriers. Okay. So if we put the sperm on this side, right? Let's say this one, and then the eggs are on this side. Okay. So you could see that you could see that uh, every time that the dominant allele is present here, okay, or if it's a carrier, like like so big A, small a, right? Because they're heterozygous, the phenotype is not present. Okay. However, because this trait is basically a recessive condition, when both recessive alleles are basically uh, present, then you will get the albinism phenotype. Okay. So if a recessive allele that causes disease is rare, so if it's a rare thing that causes a disease, it's gonna be unlikely that the two carriers will meet and mate because it's a rare recessive allele. Okay. However, people with related blood, which we call consanguineous, hopefully I'm saying this correctly, mating um, between close relatives, they increase that chance that both parents of a child carry the same rare allele, right? Because of blood similarities. Okay, so we're gonna talk about cystic fibrosis a little bit. So it is the most common lethal genetic disease. So it's inherited, okay, in the United States. And it strikes 1 out of every 2,500 people of European descent, okay? So cystic fibrosis allele results in a defect or absent in the transportation in a chloride transport channel that's in the plasma membrane. So remember, the plasma membrane and the cells have basically channels, right? And one of them basically is a chloride transport channel. So if that channel is basically bad or defective, such as in cystic fibrosis, what are you gonna expect to have? A buildup of chloride because it's a chloride transport channel. Okay. So due to this, people with cystic fibrosis will have symptoms like mucus buildup, right? Um, and malabsorption of nutrients in small intestine and things like that. Okay, so if it's untreated, it can lead to death by the age of 5, unfortunately. Okay, so but thankfully in the United States, with care and medication and all of that, um, patients can survive well into their 40s. Okay. Now, talking about the sickle cell disease, so that's a different disease, okay? So that affects 1 out of 400 African Americans, okay? So that is caused by a substitution of a single amino acid in the hemoglobin protein in red blood cells, okay? So it affects red blood cells. So what do we know about red blood cells? The red blood cells carry oxygen, okay? So we'll see what that looks like for these individuals. So in the homozygous individuals, so that means what? If the genes are both alleles are the same, right? Homozygous, okay? All hemoglobin is abnormal. So that is basically the worst case scenario, okay? Symptoms include physical weakness, pain, organ damage, and even paralysis, okay? Now, heterozygotes of this condition, so that means one of the allele is like an okay allele, and the second one is basically the defective or sickle cell, has a sickle cell trait, okay? These individuals are healthy, right? Because, they have a lower amount of gene presentation. Okay, so they suffer some symptoms, but not the whole thing, like, a homozygous individual. I wanted to mention also that this genotype, okay, the heterozygotes are less susceptible to the malaria parasite. So there's a benefit to that, okay, an advantage, in places where malaria is prevalent. Okay, so this is showing you the homozygote, okay, and then the heterozygote. You see how they're like similar here, the alleles, and then they're basically one is normal and then one is defective in the heterozygote. Okay, so one will be able to kind of have both a normal and sickled red blood cells, while this one, the homozygote, will be completely sickle cell disease. Okay. Okay, so now let's talk about just the dominantly inherited disorders. So that means these disorders are due to a dominant allele, not a recessive. Okay, so dwarfism. Okay, so it is basically due to a dominant, not a recessive allele. Okay, so in order for dwarfism to show up, okay, this phenotype to show up, the dominant allele has to be present. Do you see that? Not the recessive. The recessive is normal phenotype. So dwarfism is called achondroplasia. Okay. Also, there's some diseases that have like a timing of onset, so they do not show up immediately, but they show up like later on in individual's life, such as Huntington's disease. Okay, so that's a degenerative disease of the nervous system. This disease shows up in individuals in their 40s, 35 to 40s basically. So their nervous system will start basically deteriorating, and obviously nerves cannot be repaired, and so that is sometimes basically fatal. Okay, so there are tests, genetic tests, obviously nowadays, because of like the advance of science, that is able to examine, family members, and figure out if the Huntington's disease is actually present, and people can be tested for, and basically figure out if that gene will basically be expressed, and phenotype presentation will appear later on down their life. Okay, so um, a lot of diseases obviously can also have genetic and environmental components. So both components contribute to a disease showing up, basically. Okay. So in the case of also, conceiving in babies, also genetic testing can reveal a lot of genetic disorders, and can basically tell parents even before conceiving, you know, um, whether a certain disease will basically show up, okay, or not. So there are a lot of genetic testing that can be done at any stage, basically. Even during pregnancy, they could basically extract the fetal fluid and all of that, and I'm going to show you that actually. Yeah, right there. The um, chorionic tissue as well. Okay, to figure out if basically um, a baby is basically carrying a disease or is going to develop a disease. Okay, so let's see here. So if both members of the couple, right, had a sibling with a recessively inherited illness, both of their parents were carriers, right? That is why the sibling has a recessive inherited illness. Okay, so that means that the parents were definitely carriers, because eventually the heterozygotes are gonna basically give out a homozygous recessive individual, as we knew already from all of these things we've been studying. Okay. So that would be the 1/4 or 25% chance, right, having the recessive illness. Okay. Okay, so in the case of babies, as I mentioned, we have the amniocentesis, which is basically the liquid that bathes the fetus. Um, we can remove it and basically test that out for genetic diseases and disorders. Okay, there's also the chorionic villus sampling. It's called CVS. Okay, it's a sample of the placenta that can be basically removed from mother. Okay, and it can be tested to figure out if also there's any type of disease. And these are basically how each one is tested. So amniocentesis is tested with a needle by being inserted inside um, the placenta, right? And then the amniotic fluid is basically withdrawn in that needle, and then it can be tested, and the karyotyping will be done, obviously, which is where scientists and lab technicians can see basically the chromosomes of the individual. Okay, chorionic villus sampling is also done by basically getting a sample of the placental sampling, and that is done by a suction tube inserted through the cervix of the mother. Okay. Okay, so also some genetic disorders, they can also be detected by simple tests. So one of them is phenylketonuria, PKU. So that is a recessively inherited disorder that occurs in one of every 10,000 to 15,000 births in the United States. And that should be it for this lecture. So now this one's a long one, and it just requires repetition and memorization of things. So please go ahead and sorry about that. Study these things, and I'll see you on the next chapter. Have a good one.